Welcome to this custom-tailored deep dive. We're so thrilled you could join us today because, well, you've sent over a stack of sources that frankly kept me up way too late last night reading.
Yeah, it's a dense stack for sure.
It really is. We are looking at two highly technical papers from early 2026 that outline what I can only describe as a massive leap forward. Our mission today is to explore the dawn of end-to-end automated science.
Which is, it's a huge claim, but the data actually backs it up here.
Exactly. So first, we have a February 10, 2026 paper from Isomorphic Labs. And this one details IsoDDE, which is an AI designed to model and create revolutionary drugs from scratch.
Right.
And then right next to it, we have a March 2, 2026 paper introducing CEPROAGENTS. This is an AI multi-agent system that actually tackles the physical manufacturing of those exact chemicals, but at an industrial scale.
It is such a brilliant pairing of sources because it highlights this massive, honestly, often ignored chasm in scientific discovery.
The gap between the digital and the physical.
Exactly. We hear all the time about, algorithms discovering a miracle molecule in a digital simulation, and that's great, but there is an entirely different universe of difficulty in taking that digital molecule and actually designing the physical plumbing.
The actual factories.
Yes, the massive reaction vessels, the thermal integrations, everything required to mass produce it safely. Today's deep dive bridges that exact gap. We are looking at the software that invents the molecule, paired directly with the software that engineers the factory to manufacture it.
Okay, let's unpack this by starting on the molecular side. The Isomorphic Labs Drug Design Engine, or IsoDDE. Now, if you are following this space, you already know AlphaFold 3 was a monumental breakthrough for predicting static molecular structures.
Absolutely foundational.
But your source highlights a really persistent bottleneck with those older models. They genuinely struggle when they were forced into completely unexplored regions of molecular space. If a structure was too far outside their training data, their accuracy just, well, it plummeted.
It fell off a cliff.
Right. And IsoDDE was built to shatter that ceiling. The paper points to the runs and poses benchmark to prove it. On the absolute hardest part of this test.
The 0 to 20% similarity bin.
Yes, the 0 to 20% bin, where the AI is basically looking at alien geometry it has never encountered before. In that dark territory, IsoDDE achieved a 50% success rate.
Which is staggering.
It's more than double the accuracy of AlphaFold 3 in that exact same test.
And to really appreciate what a 50% success rate in that specific bin actually means, we have to look at how proteins behave in the real world.
Because they aren't static.
No, not at all. For a long time, the standard way to visualize drug interactions was the old lock and key model. You have a rigid lock, you insert a rigid key.
Right, very simple geometry.
But biology isn't rigid, it's incredibly dynamic. We are used to induced fits, where the protein actually shifts and molds itself to accommodate a drug as it gets closer.
Like a baseball glove catching a ball.
Exactly, the glove closes around it. But IsoDDE is tackling something much, much harder than that. It's tackling cryptic pockets.
Right, and cryptic pockets are a whole different beast. It's not just the lock changing shape. It is like looking at a solid brick wall, just a flat surface where a doorway is completely plastered over and invisible, and it stays invisible until you lean on the exact right mark, and suddenly a hidden door swings open.
You hit the nail on the head. These pockets are completely flat. They are indiscernible on the protein's surface until the specific ligand arrives.
And that's why AlphaFold 3 couldn't find them?
Precisely. Older models like AlphaFold 3 fundamentally could not predict these hidden doors because they relied heavily on recognizing patterns from their training data. If the open pocket wasn't in the database,
The model just didn't see it.
right, it was blind to it. IsoDDE, however, understands the underlying physics and spatial energetics so deeply that it can actually deduce that a pocket will form even if it has never seen it happen before.
And the paper dives into a fascinating case study to demonstrate this, using the PDB structure 8EA6. I was actually hoping we would get to this 8EA6 target because the mechanics there are just wild.
It's a perfect showcase of the tech. So in this specific case, the ligand acts as an allosteric inhibitor.
Meaning it doesn't bind to the main active site?
Exactly. It doesn't just block the main site. It binds to a completely different secondary location on the structure to force a conformational change from a distance.
It's like pulling a lever in the hallway to lock a door in the kitchen.
Great analogy. The ligand forces its way between two NKG2D protein chains. It wedges them apart and creates a pocket that simply did not exist in the unbound state. And IsoDDE predicted this precise allosteric wedging action purely from the foundational chemistry.
And the paper pushes that even further with the cereblon, or CRBN, case study. Now, cereblon is a major target in targeted protein degradation. IsoDDE was tasked with finding a novel cryptic site on it. But here's the part that genuinely blew my mind.
The sequence-only prediction.
Yes, the AI accurately found this hidden site using just the bare protein sequence. It didn't even know the identity of the ligand it was supposed to be designing for.
It essentially looked at the foundation blueprints of the house and pointed out where a secret room could logically be built.
Exactly. And when researchers finally introduced the specific ligand, a molecule called SB405483-IsoDDE predicted its exact binding pose with a tiny 0.33 angstrom error.
And just to put an angstrom into perspective for a second, that is 1/10,000,000th of a meter. We are talking about true atomic level precision here.
It's unfathomably small.
It is, predicting the exact orientation of a complex molecule inside a hidden pocket that the AI deduced from a bare sequence tree. I mean, it's nothing short of revolutionary.
But the source doesn't just stop at small molecule drugs. It pivots into biologics, specifically antibodies. And the paper focuses heavily on the CDRH3 loop.
Oh, the CDRH3 loop, the absolute bane of structural biologists everywhere.
Right. For those who might just be catching up on antibody modeling, the CDRH3 loop is essentially the hypervariable finger of the antibody. It's the part that reaches out and actively grabs the target antigen.
And it is incredibly floppy.
Extremely floppy. It's constantly changing shape, which makes it a complete nightmare to model computationally.
It really is the Wild West of structural biology. Because that loop is so flexible and unstructured, traditional AI models completely fail to capture its actual binding posture. And if you can't model that loop, your virtual antibody design is essentially useless in the real world.
Which brings us to a highly technical, but honestly pretty funny, failure of the older models mentioned in the paper. They were looking at a specific target, PDB-ID9FZD.
Right, the nanobody target.
Yes, a nanobody trying to bind to an OMPA membrane protein. Now, membrane proteins are notoriously stubborn targets to begin with. When AlphaFold 3 tried to solve it, it scored a flat 0.00 on the .Q metric.
Just a total failure.
Absolute failure. And the reason why is fantastic. AlphaFold 3 literally placed the nanobody on the wrong side of the cellular membrane.
It's the molecular equivalent of trying to unlock your front door while standing inside your neighbor's living room.
That is exactly what it is. It completely lost all spatial awareness.
But when they ran that exact same 9FZD target through IsoDDE, the results were drastically different. IsoDDE achieved a 0.943 .Q score.
So it placed it on the correct side of the membrane.
On the correct side. And furthermore, it modeled that incredibly difficult CDRH3 loop with a 0.94 angstrom accuracy. It understood the spatial constraints of the membrane environment in a way the previous generation simply couldn't grasp.
So we have an AI that can perfectly predict how a molecule fits into a pocket. But fitting is really only half the battle.
Yeah.
I mean, if I put a key in a lock, but it's so loose it just falls out a second later, the drug won't work. We need to talk about binding affinity.
What's fascinating here is how IsoDDE approaches that exact problem. Binding affinity is basically the measure of how tightly the drug holds onto the target once it's actually inside the pocket.
The grip strength.
Exactly, the grip strength. Historically, predicting this required computationally massive physics-based methods. We're talking about programs like FEP Plus or OpenFE.
And those take a lot of juice to run.
An unbelievable amount. These programs run molecular dynamics simulations. They calculate the physical forces between atoms femtosecond by femtosecond. They are the absolute gold standard for accuracy, but the computational overhead is astronomical. They require enormous amounts of time and dedicated supercomputing power.
Are you saying IsoDDE is replacing those heavy physics simulators? Because that seems like a massive leap for a deep learning model to make on its own.
The data in your source suggests it is doing exactly that. Across rigorous blind tests, things like the CASP16 competition and the Chembol 35 benchmark, IsoDDE is matching, and in some cases beating, those computationally heavy physics-based methods at predicting binding affinity.
Without the supercomputers.
Without the massive overhead, yes. Because it's using advanced deep learning architectures rather than brute force physics simulations, it evaluates the binding affinity in a fraction of the time. It has internalized the rules of chemistry so thoroughly that it can instantly score the grip strength of the molecule without needing to simulate every single atomic vibration.
Okay, here's where it gets really interesting and where we transition to the second paper in your stack. Let's say IsoDDE does its job perfectly. It finds a cryptic pocket on a cancer-causing protein. It designs a highly complex small molecule that fits with atomic precision. It predicts a rock-solid binding affinity.
We have the cure.
We have the digital blueprint for a cure sitting on a hard drive. But we need 1 million gallons of it. We need a factory. We need reactors, distillation columns, specific thermal profiles. Enter CEPROAGENTS.
Yes. This is where the theoretical hits the concrete. Designing a chemical plant to mass-produce a novel molecule is incredibly complex. Traditionally, you need a room full of specialized human engineers,
Chemical engineers, process engineers, safety analysts.
all working together for months just to design a functional process flow diagram. CEPROAGENTS automates this entirely by creating a virtual engineering boardroom. It is a multi-agent system divided into 3 specific cohorts.
Let's walk through those cohorts for the listener. First, you have the knowledge cohort. These are your researchers. They autonomously scour the web, pull patents, read internal database rules.
Gathering all the constraints.
Right. Then, the concept cohort. These are your designers and draftsmen. They take the research and start connecting the virtual pipes, drawing up the actual process flow diagrams, or PFDs.
The actual layout.
Exactly. Finally, the parameter cohort. These are the mathematicians who take the flow diagram and run the heavy thermodynamics to optimize the pressures, flow rates, and temperatures.
It's a full
It is, but I have to play devil's advocate here. We are talking about large language models, and LLMs hallucinate. In creative writing, a hallucination is a funny quirk. In chemical engineering, if an AI hallucinates the pressure limits on a distillation column, a factory explodes. How can we possibly trust an AI to design a high-stakes industrial facility?
That is the most critical question you could ask, and it is exactly what the CEPROAGENTS architecture was built to solve. The genius of this system is its hybrid architecture. It doesn't just let an LLM loose with a blank canvas and hope for the best.
It puts guardrails on them.
Heavy guardrails. Each cohort operates using 2 distinct components, a chat group and a structured workflow. The chat group is where the AI agents engage in collaborative reasoning. They debate ideas, synthesize data, brainstorm just like engineers around a whiteboard.
The creative side.
Right. But that creative deliberation is strictly governed by the workflow component, which is a rigid, deterministic execution path. By forcing the creative output of the chat group through the mathematical reality of the workflow, the system actively filters out those dangerous hallucinations.
And your source details a massive benchmark called CEPROBENCH, which they used to test this exact system. They took the top LLMs of early 2026, Gemini 3.0 Pro, GPT 5.0, Claude 4.5 Opus, DeepSeek V3, and Qwen 3.0 Max, and ran them through a gauntlet of industrial design challenges. Let's look at the knowledge cohort first with the isoprene production case study.
So isoprene is a vital industrial chemical used in synthetic rubber. The AI agents were given a blank slate and told to figure out the best way to produce it at scale.
Go.
Exactly. The web search agent went out and identified that a company named Sinopec currently dominates the market with a 70% share using a specific extraction method. At the exact same time, the internal database agent queried the system rules and noted that for any new plant to be economically viable, the solvent recovery rate had to exceed 99.5%.
So they're essentially doing competitive market analysis and complex regulatory compliance at the exact same time.
Precisely. And then the reporting agent took that diverse stream of data and synthesized a strategic recommendation. It didn't just spit out facts, it proposed a specific process called extractive distillation.
Which does what exactly?
It involves adding a third solvent to alter the volatility of tightly bound liquids so they can be separated more easily. The agent calculated that using this specific method would boost marginal profits by 15%. The AI didn't just solve an engineering problem, it built a highly lucrative business strategy from scratch.
That is genuinely impressive. But my absolute favorite case study from the paper highlights the concept cohort because it directly answers my earlier question about the hallucinations. The AI was tasked with designing a facility for 103 propanediol.
Right, the diagram challenge.
Yeah. The design agent got to work drawing up the flow diagram, but it made a critical error. It hallucinated a piece of equipment called a membrane separator. Now, in the strict, standardized vocabulary of this engineering ontology, a membrane separator does not exist. It was a clear vocabulary violation.
And in a traditional single-agent LLM setup, the model would have confidently continued building the factory around this imaginary machine, rendering the entire blueprint completely useless.
Or dangerous.
Or dangerous, yes. But because CEPROAGENTS uses a multi-agent adversarial loop, The correction agent immediately stepped in. It acted as an auditor, it flagged the vocabulary violation, challenged the design agent, autonomously replaced the hallucinated machine with a structurally sound gas-liquid separator.
It fixed its own mistake.
It did. And while it was auditing the diagram, it actually noticed a flaw in how the hydrogen recycle loop was routed, and it optimized that pathway as well on its own. It's a self-policing system.
So the concept cohort successfully draws a legally and physically sound factory diagram. Now it hands the blueprints over to the parameter cohort for the heavy lifting. The paper highlights an aromatics purification separation process.
This is a tough one.
It is. For the listener, aromatics purification involves separating highly complex chemical mixtures, which requires incredibly precise temperature and pressure controls across massive industrial columns.
This is where the economic viability of the entire project is decided. Using Gemini 3.0 Pro as the reasoning engine, the parameter cohort initiated an autonomous closed-loop refinement cycle, hooking directly into an industrial simulator. The initial mandate was pure quality control. It had to hit a 97.2% purity target for the final chemical.
It did it.
By the second iteration of its simulation, the AI had adjusted the thermal parameters and hit that exact purity target perfectly.
But it didn't stop there.
It didn't. The moment the purity constraint was satisfied, the internal analyst agent dynamically shifted its objective function. It moved from quality control to aggressive cost-cutting.
How so?
It began tweaking reflux ratios, which is the amount of liquid returned to the top of the distillation column to ensure separation and optimizing feed stages to reduce the overall energy load. Over the next 4 iterations, it systematically dropped the operating cost of the facility from $36.6 per hour down to $28.9 per hour. That is a 21% reduction in energy and operational costs, achieved completely autonomously, while perfectly maintaining that strict 97.2% purity line.
A 21% reduction in energy costs at an industrial scale is literally the difference between a project being greenlit or being canceled entirely. It is massive. So how did the different LLM models actually perform when pitted against each other in this CEPROBENCH showdown?
The benchmark revealed some fascinating quirks about the personalities, if you will, of these models when applied to rigorous engineering. Gemini 3.0 Pro definitely took the crown for the comprehensive score. It demonstrated highly decisive convergence behavior.
Meaning it knew when it was done.
Exactly. When it found the optimal mathematical solution, it recognized it, locked it in, and terminated the search quickly, which is incredibly efficient for computational resources.
It knew when to put the pencil down. What about GPT 5.0?
GPT 5.0 was also highly capable, but it suffered from what the researchers called an over-exploration tendency. It would treat a deterministic math problem almost like a creative writing prompt.
Oh, interesting.
Yeah, it would find a highly optimal solution early on, but instead of locking it in, it would burn through its computational budget, nervously checking and rechecking slightly altered parameters, leading to redundant energy consumption.
In Claude 4.5 Opus.
Claude showed a very specific bias toward maximizing chemical yield above all else. It would push the plant to produce the absolute maximum amount of product, but it occasionally struggled to balance that high yield against the rising energy costs as elegantly as Gemini did.
So what does this all mean for you, the listener? Let's take a step back and synthesize the sheer scale of the journey these two sources just took us on. We started in the microscopic, invisible world of cellular biology. We looked at IsoDDE, an AI that can scan a bare sequence of proteins, deduce the existence of a cryptic pocket that is currently plastered shut, design a completely novel drug to wedge that pocket open, and calculate the exact atomic grip strength of that interaction without relying on massive physics simulators.
It effectively digitizes and compresses decades of trial-and-error laboratory chemistry into just hours of computation.
Exactly. And then we zoomed out to the macro scale of steel, concrete, and industrial thermodynamics. We looked at CEPROAGENTS, an AI boardroom that can analyze global market shares, debate process flow diagrams, police its own hallucinations, and mathematically optimize the exact reflux ratios needed to build a profitable industrial-scale facility to manufacture that exact drug.
The ultimate takeaway from these early 2026 papers is that the primary bottleneck in scientific advancement is no longer raw human imagination.
No, the bottleneck has been computational orchestration. Our ability to manage the overwhelming multidisciplinary complexity of taking an idea from a digital screen to physical reality. And that bottleneck is evaporating right in front of us.
If we connect this to the bigger picture, there is a fascinating and frankly daunting implication hiding in these sources that we haven't touched on yet. Right now, IsoDDE designs the drug, and CEPROAGENTS designs the factory. They are brilliant, but they are isolated tools. Human scientists are still the ones passing the baton between them. But think about what happens when these systems are integrated into a continuous autonomous pipeline.
Oh, wow.
An AI identifies a new viral target, passes it directly to IsoDDE, which invents the molecule, which instantly hands the chemical properties to separate agents, which spits out the blueprints for the manufacturing plant.
It sounds incredibly efficient, but it also sounds like a regulatory nightmare.
That is exactly the point. Consider the FDA or global regulatory bodies. Their entire framework is built on human oversight, clinical trials, and auditing the physical engineering of manufacturing plants. How does a regulatory agency certify a completely novel drug and the highly complex factory designed to build it when both were engineered entirely by an AI to AI dialogue?
How do you even inspect that?
You might not be able to. If no human engineer fully understands the millions of mathematical micro decisions the AI made to optimize that distillation column, how do you audit it? We are rapidly approaching a reality where our capacity to invent and manufacture outpaces our regulatory framework's ability to actually comprehend the science being used. The technology isn't just accelerating discovery, it is fundamentally challenging how we verify reality itself.
That is a phenomenal and slightly terrifying thought to leave on. How do we regulate an intelligence that engineers faster than we can audit? Thank you so much for sending these incredible sources our way and for letting us take this deep dive with you. Keep questioning the future, and we'll catch you on the next one.